Hey, hello everyone. Good morning, good afternoon, good evening, depending on where you are. Thank you so much for joining us today. We will be learning from Dr. James Westrip and Dr. Anders Dahlberg about red listing fungal species. I'm Katia Canteiro, the Plants and Fungi Conservation Coordinator at the Global Center for Species Survival. Just very briefly, this is a partnership between the Indianapolis Zoo and the IUCN Species Survival Commission. And basically, our job is to support the various specialist groups that are part of the IUCN SSC network and the amazing work they do to protect species around the world, including the work that is being done by the Aquatic Fungi specialist group. You can see we are a team of about 9 people. We have different coordinators covering different remits. And now I will hand over to my co-host, Jennifer Anderson, to present about their projects. Thank you.
Thank you, Katia. Hello, everyone. My name is Jenny Anderson, and I have the pleasure of being the project coordinator for the project FUNACTION, where we are a multinational consortium working together towards the conservation of aquatic fungi. Today, our consortium is 25 people strong, but already by next week, we grow to 27 people. So this is a really dynamic and exciting time for us working with aquatic fungi. Two words about our project. We are working towards the conservation of aquatic fungi, as I said, and we're doing that through developing the foundational knowledge that we need about the distributions and the drivers of those distributions for aquatic fungi. We're translating that knowledge into conservation actions, and we're communicating about aquatic fungi and about the conservation of aquatic fungi with as many of our stakeholders as we can, and this includes through webinars like this today, and through other kinds of engagements like publications and so on. So please watch us on our online platforms, or be in touch with any of us. We'd love to talk to you about conservation of aquatic fungi. And I think now we get to hear from Isabel Fernandez. Isabel?
Thank you, Jenny. I'm introducing just very briefly the IUCN Species Survival Commission Aquatic Fungi Specialist Group. This group was created towards the end of last year, so we are very recent. As we say here, we can see our vision is to develop the scientific and coordination basis for the first conservation assessment, planning, and action efforts for aquatic fungi. There are more than 6,000 species of aquatic species described, so aquatic fungi species described, between marine and freshwater species. But so far, none of these species has been assessed for its conservation statuses. And so we are very, very looking forward to learn from 2 world experts on red listing for fungi and to see how we can start working towards applying these assessments to aquatic fungi. And with that in mind, just to briefly explain a few things for the webinar today. So we will have Dr. James and Dr. Anders, and after each presentation, we will have some minutes for question and answers. So please, if you have any questions for our speakers today, make sure you write them on the Q&A option. With no further delay, I will start introducing our first speaker of today, which is Dr. James Westrip. Dr. James is a program officer in the IUCN Red List unit. He has spent the last 8 years working on the IUCN Red List of threatened species. Dr. James completed his PhD on evolutionary biology at University of Edinburgh. And he spent 3 years at the BirdLife International working as part of their team for the Red List Authority for Birds. And since 2019, he has been within the IUCN Red List unit, working as the contact point for fungal red list assessments, in addition to working with very other taxonomic groups. Dr. James sits on the Red List Technical Working Group as the fungal representative, having formerly sat in the similar role for representing birds. we are very looking forward to hearing from you, James. So I hand over to you. Thank you.
Thanks very much, Isabel. So I'll just share my screen out and then. Hopefully everyone can see my presentation. So thank you very much again, Isabel. I don't really have to do my introductory slide because you've pretty much said everything I needed to say there. So thanks very much. So I'm just going to take everyone through a very brief introduction to what the IUCN Red List is itself. So this won't be very much about fungi itself, because that will be more what Anders is going to talk about after me. So the IUCN Red List of Threatened Species is a way of quantifying extinction risk for species. So we try to use it as a barometer of life that takes the pulse of the planet, and it uses a set of categories and criteria that gives an objective assessment of the extinction risk that species face. And it uses a methodology that's comparable across taxonomic groups. So it can be used for fungi, for invertebrates, for vertebrates, plants. Anything that's not a single-celled organism can be used in this methodology. So to do this, we use a range of criteria to give a final red list category. And here on the right of the screen, we have the strict set of categories that we use at the global scale. So theoretically, every single species can fall into this system, because anything that's not been evaluated would be called not evaluated, though we don't publish not evaluated species on the Red List website. We use a system that allows us to designate species within one of these three threatened categories: critically endangered, endangered, or vulnerable. So as we're moving up the scale, up the page, that's a higher extinction risk. So critically endangered species are more at risk of extinction than endangered, and endangered are more at risk of extinction than vulnerable. We have a set of quantitative thresholds for each of these categories, but if something approaches those thresholds but doesn't actually meet them, then the species would be called near threatened. And for any species where there's such an amount of data that we can see that it wouldn't even approach those threatened thresholds, then we can call the species least concern. Any case where there's so much uncertainty in the data that the species could be anything from critically endangered to least concern, then we would call the species data deficient. Obviously, right at the top are the species that are extinct already or extinct in the wild. So they're extinct in a wild setting, but only found in a collection as a living specimen. So in a botanic garden, for instance, or in a zoo. It's very difficult to say when a species is actually extinct, because the absence of evidence isn't necessarily evidence of absence. And I won't go into the details of how we do that, but anyone who is interested in finding out what kind of thresholds we use, I'd suggest looking at the Butcher et al. 2018 paper that I've highlighted below. So to get to those categories, I've mentioned these quantitative criteria, and they have a lot of supporting information that means that what we're plugging into those criteria are parameters that can be called symptoms of extinction risk. So something like population size. The smaller a population size, the greater the risk of extinction. And population trend, so a decreasing population size has a greater chance of going extinct than something of the same population size that's stable. Geographic range, so something with a smaller range is at a higher risk of extinction potentially than something with a larger range. And also what threats a species faces. So if a species is facing a lot of very severe threats, then it is probably going to be at a higher risk of extinction than something that faces no threats at all. And all of these were laid out in the original paper for the starting way we look at these quantitative criteria in the Mace et al. Paper from 1992. Now, a red list assessment includes all of this, but it also includes a lot of other information. So you'll see if you read any assessments that there's lots of information on habitats and conservation actions and research that's going on a species. So it tries to build a bigger picture of everything that we know about a species. So pretty much the most important thing we have when we're starting, or need when we're starting a red list assessment, is good ecological data. Now, I'm going to give a slight warning to everyone that the next few slides will look like they've got a lot of information, but obviously I'm going to try and explain the general themes that are shown in the slides rather than go through every single tiny bit of information. So don't be afraid of everything that appears on there. I'll be trying to just skim through and show you actually an introduction to what the data is on there. So we use 5 different criteria to assess species, and the first is criterion A, and this looks at the rate of decline of a species. So as the general themes that you'd be wanting to look for, we have these different thresholds that are set out. So thresholds for critically endangered, for endangered and vulnerable, and they're the starting blocks that we'll be looking at. We do have these 2 different parts, different thresholds. So A1 is a very specific case where the causes of a reduction are known, they are clearly reversible and understood, and they've ceased. So all of that needs to have stopped. And theoretically, then those species have once it's stopped and we know what's causing it, and we know we can reverse it, theoretically, that species would be in a better position than something where we don't have all of those 3 ticked. So we use these different thresholds between A1 and A2, A3, A4 because of that. And to figure out these, we may have the direct data that shows we've got X amount of population that's disappeared over a certain length of time, but we may not. So we can use other pieces of information. So we may know that the range extent has declined by a certain amount, or we may have information about declines in the amount of catch there is for something like a marine fish. And we use that data to create a estimation or suspicion over a rate of population decline. Now, something that's really important in criterion A is this little bit right at the top. So measured over the longer of 10 years or 3 generations. And this is a standardization that we use that allows this criterion to be comparable for anything from a giant redwood all the way down to a fruit fly, because they obviously have very different biologies. So if we imagine 2 species with exactly the same population size, exactly the same range, exactly the same threats, and they've both undergone a reduction of exactly the same percentage in exactly the same period of time, both those 2 species would not necessarily end up in exactly the same category based on this system. Because if you've got one species that reproduces really quickly, so we'd say it has a short generation length, then theoretically it would be able to replace all of those lost individuals within a short period of time if the threat were to stop. On the case of the other species, it could reproduce very slowly and so have a very long generation length. And so it could take a very long period of time for it to replace the lost individuals. And so the one that reproduces slowly should be at an increased risk of extinction compared to the other one that reproduces very quickly. Now, how would we put this together to make an assessment under criterion A? So if we know that there is a 30% decline that's happened in the past and it hasn't stopped, then we would call the species vulnerable under criterion A2. If, on the other hand, there's going to be a 50% or more reduction in the future, then it would be endangered under A3. And now a group of species that have quite a lot of assessments under criterion A are the great whales. After criterion B, again, after criterion A, sorry, comes criterion B. And again, you can see here all of these different threshold values that we set out to assess a species: the critically endangered, the endangered, and the vulnerable. To assess under criterion B, we look at the geographic range of the species. So we start off at the top looking at extent of occurrence and area of occupancy values. To be listed under B, you can trigger one of these or both, it doesn't matter, but they do have very specific and technical meanings, EOO and AOO, but the little diagram at the bottom hopefully will explain a little bit of that. So if you have a known range of a species, which is all of these differently colored, or not differently, all of these black blobs within the green background. So the black blobs are where the species occurs. Effectively, that would be considered the area of occupancy. If you were to draw a minimum convex polygon around all of those occupied areas, it is that total area within the bounds that would effectively be this blue square. That is the extent of occurrence, because the extent of occurrence is more a measure of the spread of the species. So once we've got those measures, we would then look at other bits to figure out what else goes into the criteria. And we need to have at least two of the following three conditions. So whether there's a small number of locations where the species occurs, and looking at whether there's any declines or if there's any extreme fluctuations, because all of these feed into the extinction risk of the species. And we could put that together by saying, if you've got an extent of occurrence of under 100 kilometers squared, and it's found at only one location, and there's a continuing decline in the number of mature individuals, then a species could be called critically endangered B1 ABV. And this cichlid fish from Lake Malawi is an example of that. Following on for criterion B is, of course, criterion C, and this looks at small population sizes that are in decline. So again, we have here our critically endangered, endangered, and vulnerable thresholds. And we start off by looking at whether there's a population size that would fall within these boundaries. And then once we've done that, we need to know whether there's a decline. And if we know that there's a small population size and a decline, we then start to look under the sub-criteria C1 and C2 to see whether it does deserve a listing under criterion C. So again, we have here generation lengths for C1. So looking at whether there's a small population which has undergone a certain percentage decline within a set period of time, but it doesn't have to be the case that that's met. Instead, we could look at the structure of a population or whether it's undergoing extreme fluctuations. An example of a species that's listed under criterion C is the Florida scrub jay. And so it's got a small population size, which is declining by a set percentage over 3 generations. And it also has a fragmented population. So it's called vulnerable C1 plus 2AI. Now, I'm going to note that this is an older assessment because it's been reassessed since 2017, but there's something really interesting about that 2017 assessment that I will refer back to in a few slides' time. Now, the final two criteria we use are criterion D and criterion E. D looks at very small population sizes or highly restricted populations. So again, we have our threshold values, and for critically endangered and endangered, we only look at the case where there's tiny populations, so fewer than 50 mature individuals and fewer than 250 mature individuals. But for vulnerable, there are two different ways that it could be triggered. So firstly, it could use D1 if there's just a small population size. But for D2, there's the chance for it to be listed as vulnerable as well if it has a very restricted range, and there's also a plausible threat that's going to drive the species to critically endangered or extinct in a very short period of time. And a group of species that have been assessed under criterion D quite a bit are the cycads. The final criterion E, is quantitative analyses, and these use a lot of ecological data where they're fed into modeling to find the probability of a species going extinct in the wild. It's very rare for a species to be assessed under criterion E. It's increasing more now that people are trying to do them, but generally there's so few that are assessed under that because there's so few species that have sufficient data to produce a good enough model that our standards and petitions committee would accept. So I don't have any examples for that to show, sorry. Now, one thing you need to do is not just to think about a single criterion, and if it triggers that, then that's it, finished. So I'm bringing back the Florida scrub jay, and its 2017 assessment was not just the C1 plus 2AI that I mentioned earlier. It was vulnerable under criterion A and C. So if we just stopped after going one criterion by another, we'd have only had it vulnerable under A. We wouldn't have also figured out that it was also vulnerable under criterion C. So we need to take every single step and look at every single criterion that we have the available data for. You could say that that's not really a problem in the case of that species, because if we just gone and looked at criterion A, found it's vulnerable, nothing's changed based on that. It would still be vulnerable as the final category. But what if you had a situation like this, where you have a species vulnerable under A, endangered under B, critically endangered under C, endangered under D, and then we didn't have the data for criterion E? Well, in this case, the species should be critically endangered under criterion C. So if we just stopped after criterion A because we triggered a threatened category, we would actually be not fully recognizing how bad the situation is for a species, and we would underlist it. So that's why we need to go through every single criterion when we have the data available. So most of the time, assessments are conducted or reviewed by members of the IUCN SSC network, the Red List authorities, and Red List partners, as well as experts on IUCN-led projects. But assessments can be done by anyone who has sufficient knowledge, and they can be submitted for consideration to the Red List unit. So you just need to do the training, which is free online, and then you'd have an idea of how to do the assessments based on the full guidelines, and then you could attempt to do an assessment of a species. So we've been doing this again and again for a very long time, and these numbers are very much hot off the press because it was only yesterday that we did our latest release, and these are the current numbers for species totals in every single category. So we have just over 900 species that are listed as having gone extinct, and we use a cutoff date of 1500. So these are species that have gone extinct or have been extant since 1500 and then gone extinct since then. So all of these species are things that are extant since 1500. There's 81 that are considered extinct in the wild, and then we hit our threatened categories of about 10,000 that are critically endangered, 18,000 endangered, and about 17,250 are vulnerable. Just under 9,500 species have been assessed as near threatened, and over 85,000 assessed as least concern. There's an increasing percentage being considered data deficient, so we just have under 22,000 that are considered data deficient, and this all gives a total of just over 163,000 species that have been assessed and published on the Global Red List. Now, anyone who is very good at mental arithmetic will add all of these up and will not come to 163,040. That's because there is another category out there called lower risk conservation dependent, which is not in the latest versions of the categories criteria. It's an old category which we're phasing out slowly. So that's why the numbers don't completely match up. So the ecological data we've been using to make assessments doesn't just highlight the species that are threatened with extinction, but also lets us know why they're threatened. So there could be a range of different threats depending on what the species is. So hunting is a threat to lots of vertebrates in particular. Logging can be a threat, not just to the trees, but to the species that live in the forests. Invasive species could be a direct threat, or direct and or indirect threat to a species. So they could be something that's killing off a native species, or they could be disturbing the habitat to such an extent to make it harder for a species to live there. But these are just a handful. They're not a comprehensively list of the threats that we use. So in a red list assessment, we can identify the species that are threatened and also the threats that are causing them to be threatened. So in theory, this should allow us to enact targeted conservation. And this is something that BirdLife International have been involved with. So albatross species are a group that's highly threatened. And one of the key threats to that is longline fishing. And the thing that has been come up with is called the hook pod. So this is a device that kind of encircles the hook on a fishing line in these commercial fisheries, and it bases things on pressure. So the albatrosses can only dive down a certain distance, but the target fish that the fisheries are targeting are below that depth within the water. So as the hook pod gets deeper, it uses the pressure to know when it's at the right depth before it opens up, allowing the hook to be available to the target fish, and should theoretically not catch the albatrosses then. And the result of using this has been a 95% lower bycatch of albatrosses in the fisheries that have been using these hook pods. And the impressive thing is there's no negative impact on the catch rate of target species, because of course we could always reduce the amount of albatrosses that are being caught, but if it reduces the number of fish that are being caught, then the fishing industry is not going to want to take it on. But positively from that, it doesn't have that impact. Another thing, if we identify species and a broad-scale threat, we can enact broad-scale conservation. And so with invasive species, you can identify with rats on islands or invasive plants that are degrading certain habitats, and you can affect a whole suite of species and help their survival by targeting these specific threats, which you know are a threat across a broad scale in a certain area. Now, that's a very brief introduction. There's obviously a lot more data and a lot more technicalities. And so if you wanted to, more information is available on the IUCN Red List website. And that's the link there if anyone wants to take a note of it. And that's the end of my presentation. So if there's any questions, please do let me know. I'll stop my share.
Thank you so much, James, for this great introduction to red listing. Actually, there are questions, and I would start maybe with one question from actually Jennifer. And she actually, we were discussing this in private, but maybe we can make this discussion public. So what about the importance of the data deficient and least concern categories? Why it is valuable to do these assessments that may end up with this designation. So what is your opinion on this? Thank you.
So data deficient for me is incredibly important. So that one more so than least concern, in my opinion, because data deficient shows where there isn't data. So as soon as data deficient could be critically endangered, it could be least concern. But if you're being ultra precautionary, this thing could be at the point of going extinct. We just don't know. And so highlighting that at such a level then says, well, we know so little about this. This could go extinct. It's a really important thing that we need to conserve it. So we need to be able to get more information so we know exactly what's going on with the species. So it can be useful as a tool to highlight where research needs to go in the future. Whether funding bodies want to listen to that or not, it's up to the funding bodies and up to the researchers whether they want to, but it's the ability to highlight where research may want to look because there's the threat that it could be critically endangered. If we just focus on the threatened ones, then we could be ignoring a huge number, because there's 21,000 data deficients. That's not as many as in the threatened, but a large proportion of those data deficient could be threatened species. So it's that kind of worry. Doing the least concerns, it's important to know the proportion of the world that is threatened. If we didn't do least concern assessments, we wouldn't know how much is under threat, because we could just be leaving. There's a huge number that are least concern, obviously, but we could just be saying, oh, they're all fine, or they're not assessed. You wouldn't be knowing whether that's there or not. Yes, it's not the most important from an extinction risk side of things, but a least concern assessment doesn't mean it shouldn't be conserved, because something could be least concern because it doesn't quite get to near threatened, but it could be an incredibly important species. And because the red list assessment, the final category is about the extinction risk, but elsewhere in there can be text about the importance of the species. Is it used or traded? Is it have a high economic importance for that? Does it have a high cultural importance for certain societies? That it all goes into the assessment. Yes, we'd just see least concern as the final part, but without publishing that it's least concern, no one would know. So that's why it's important as much as anything. We can only say and produce best reports and things like that give us this estimation of the total number of species that are at risk of extinction, because we know how many aren't at risk of extinction as well. I hope that answers.
Thank you. Katya, hand over to you.
Thank you, James. We have a question from Pratyasha about, are there any openings or opportunities for Sci artists to get involved with the IUCN Red List, especially with respect to the fungal red lists? So I'm guessing this is a scientific illustrator or artist, and how can they get involved?
So I'm not an artist. Very much, I'm a very, very poor artist. But in general, I don't know of any, because I just don't work with it. I work on the science side of things, but the best thing to do would be to send an email to the Red List inbox. So on our website, we have our generic Red List unit email, and everything that goes in there is read by members of the team, and that can then be passed on to senior members who know and may have dealt with things like this in the past and queries like that. I'm really sorry, I just haven't had to deal with anything like that before. I apologize.
Just to add to that, thank you, that if you're looking to get involved with the specialist groups in general and just not red listing, reach out to us, because there are opportunities to get involved with the specialist groups with some communication work. Thank you. Over to you, Isabel.
Thank you. We have one last question before we move on to Anders. The question is from Andreas Bruder, and the question is the following. If red listing is global, how can we deal with knowledge gaps on the distribution of certain species? For instance, in suspected but understudied biodiversity hotspots.
So I could talk for a long time. I'll try and so if it's of a species and you just don't know if it's occurring somewhere or not, there are two different approaches to take. So there's the approach where you can make an inference whether there's suitable habitat, the likelihood it's going to be there, and you can make inferences about whether it's there or not. On the other side, you can be ultra precautionary and say, we're only going to assess something exactly where we know it definitely occurs, because that's the minimum every single one of the parameters that we know it's going to be. So that's the worst case scenario. When you're doing the assessment, you usually end up somewhere in between. And the way that we do fungal assessments, which Anders will come on to, is actually a really great way of finding that in between, not going all the way to assuming everything's least concern, but not going, we've only found this twice, therefore we're going to say there's only 2 individuals. Well, you may have only looked twice. It's that kind of thing. We have a really good middle ground that the fungal assessing follows. On a broader scale, we try to build up from the knowledge we have. So at the moment, the red list is obviously very much biased towards certain groups that have been comprehensively done in terms of what we're able to build our full knowledge on. And most of those comprehensive groups are going to be in the vertebrates, because that's what things were like at the start. This is why the barometer of life exists now, to try and these targets to try and to get the red list to be a true barometer of all life, and why we're trying to do comprehensive assessments in other groups as well, so that they can feed into our analyses to figure out exactly what's going on across various places and at specific biodiversity hotspots. So at the moment, if you try to do analyses, it will probably be birds, mammals, amphibians. But in our red list index, we also have things like cycads and corals. So it's not just vertebrates, but the more assessments we can get from other groups and comprehensively assess them, the stronger that as a tool the red list will become. So the more aquatic fungi we can get on the red list, even better.
Thank you, James. This was great. And as Isabel put in the chat, we'll have time for more questions at the end. So please keep putting them in the Q&A. And it's my pleasure to introduce our second speaker. Dr. Anders Dahlberg is a professor in mycology at the Swedish University of Agricultural Science in Uppsala, Sweden. His research and teaching are focused on fungal ecology and conservation in forest ecosystems. And together with Dr. Greg Mueller, he wrote the guidelines used for fungal conservation, for fungal red listing. And he also coordinates the Global Fungal Red List Initiative together with Dr. Mueller. And he's a member of and the Red List Authority Coordinator for the IUCN SSC Mushroom, Bracket, and Puffball Specialist Group. Anders will share his 20 years experience from working with the National Red List and the decade with IUCN Global Red List. Over to you, Anders.
Thank you. I will start my presentation here. Let's see. So let's see. So does it work now?
Perfect.
Yep. No, pleasure to be here and to share some of sort of the joint experiences over the last years of red listing. And please feel free to ask a lot, but I will give a sort of an introduction of why I think it's important to do red listing, what it has meant and can mean, its significance, and a little bit about how it has been, how we conduct it. And I think this why question is very important. Why we do this? Because all of us, there are many things in life we want to do, and in order to prioritize and to allocate our time as wise as possible, I think it's important to really figure out why are we doing things like red listing, because it's enjoyable and fun and interesting, but it takes time. And I think in my perspective, I think, I mean, whatever we do, it has an anthropocentric perspective. We do it for ourselves. I mean, as I'm thinking, nature by itself doesn't care whether it will be present tomorrow or not, but we care tremendously about the species we have and the ecosystems we have. And then we can argue from different angles why we think this is important for what these species, what they do for us. And we can have, as you are well aware of, sort of insurance for how nature is working, processes are working in nature, or it can be aesthetic reason or ethical reasons that we have that we should care about those species that are present. But I think it's important to be aware of that, and also this perspective that by knowing the status and trends of species, and to ensure that we have knowledge of the species, their ecology, where they occur, what factors are disfavoring them, and how they, in ways to promote their existence, we can manage nature in a better way. It provides a tool to direct the handling of nature, hopefully, in a way that we get the nature, the biodiversity we want to have in the future. For a long time, I mean, animal and plants globally has been the focus of conservation, and I think that, in my perspective, fungi is obvious that fungi should be included as well, and it is included in many countries already and been so for many years. But it's obvious that all aspects of biodiversity that we could assess should join and complement each other to strengthen our awareness of what is going on in nature, and to help to better handle conservation together. So fungi strengthen animal and plants, and plants and animals strengthen fungi. So it's a joint effort to make sort of the best management of possible into the future. And the experience I have is that, of course, there can be conservation actions to single species or groups of species, like albatrosses, as James talked about. And that is fine, of course, for species that we, for various reasons, appreciate particularly much. But more commonly, sort of, we handle groups of species together, or I would say that the most common things to manage nature is to manage habitat. And then, in order to have a good grip of what habitats there are species that are not doing so well, to identify those and to identify what are the threats and to identify how to counteract that. The better knowledge we have of the species present in that habitat, the better we can provide guidelines and work in that direction. So think of that perspective to make joint efforts for that. Fungi will be an excellent contribution to, whether, irrespective if it is terrestrial species or species in water environment. And what I think is very good with the red listing, the IUCN red listing system, is that it's the same criteria, it's the same assessment process for any group of organisms. Which means that it's easy for decision makers and stakeholders, politicians to understand that conservation is speaking with one voice. That is sort of critical for reaching their ear and for moving conservation forwards, to have the same system. In most countries, as I'm from the Nordic country, from Sweden, where there is no the red list is not linked to legislation, and the IUCN red list by itself is not linked in any way to legislation. There are some countries that have legislation linked to their assessment, but normally that is not the case. And I think that is a strength that we have in the decision-making process, that we have the red list somewhere in between these sort of so-called soft values, where people, they like certain species, or they appreciate certain appearances, or species for various reasons. In contrast to the hard factor, where there is legislation that if a species is present, then it must be handled in a certain way. Because there are, as I see it, there are so many other ways or aspects to consider in management, in managing land, and the status and trends of species is one aspect, an important aspect, but it's not the only aspect. There are other reasons, and we have other anthropocentric perspectives on managing land and species as well. And it also makes it much more uncomplicated to assess everything, and to do that as objectively as possibly, and then in the next step make priorities of which species and for which reasons, considering other aspects, should be prioritized in the conservation work. And a very important aspect, I think, and what I have gained with the work with the red listing over the years, is that it actually forces and it stimulates very much thinking about fungal biology, about fungal population biology, how the fungi make their lives go around, how they complete their life cycles, how long-lived are they, how are they dispersed, what are their sort of population characteristics. And I think that is sort of the driving thing for me working with red listing. I think conservation is important, of course, but this has that stimulates my thinking very much. How are they live? And I hope that is doing that already for many of you, and will do some more in the future. And one aspect there, which I think is important to know, beside that, it's just this tiny fraction of the species, as you know, of fungi that are described yet. The overwhelming part are yet unknown and undescribed. It is that we, just so that you acknowledge that there's a huge diversity of life forms of fungi in the world. They can be small and they can be large, and they can be short-lived and long-lived, in the same way as we have zooplankton and whales. I mean, they belong to the animals. We have annual plants, we have very long-lived plants, and they have very different traits and adaptations for their life. It's the same with fungi. Just appreciate that. But as you know, red list is very much about the status and trend of species. And I remember when I started with doing this, I was very confused about these criteria and how that worked, and with the different letters and so forth. It was a very tricky language to get into. But in principle, it's very simple. It's about if there are common species that are declining, uncommon species that are declining, or very unusual species. Those species will be sort of sorted into the group by this assessment process to become classified as threatened or near threatened. I'll come back to that later. And as I just want to remind that national red lists are very much in use, and it's very much appreciated all over the world. And there are quite, I would say, relatively few, or quite a few, but there are about 35 countries in the world that have national fungal red lists. And some countries have had them for very many years, for 40 years. And they mean a lot for the management of fungi in those countries. On the global level, as James just mentioned, there was an update recently. We didn't really start to do red listing on the global level until less than 10 years ago. And now it is few compared to animal and plants, but still it's about 800 assessed. And 300 threatened there. And that's very much thanks to the Global Fungal Red List initiatives, which is a website where we work with physical and digital workshop. You're welcome to go into that website and have a look. Just to give a little bit of my experiences, because I have mainly been working in Sweden, where we have been red listing since 1984, so it's 40 years, and done that repeatedly. Here you see some of the data also from Finland and Norway. They have the same, a long tradition of doing national red listing. And over the years, it's become more and more fungi, so assessed. So in Sweden, we have about 6,000 macrofungi known, and more than roughly half of them has been assessed. And we have 2,500 known lichens, and about half of them are also assessed. So it's a substantial number of species that are assessed, and that is the same for quite a few countries nationally. And you see here on the pie chart there that fungi, looking on the national level where assessment have been conducted, they constitute a quite significant part of the assessed species, and therefore also means a lot for conservation actions. And that is my experiences when the red listing has been conducted for a long time, that it successfully become more and more well known and become accepted and has a significant impact. In many of these of the national countries, it is now a government document, and it is also a strength, which it is in the Nordic countries, that it's coordinated for all species groups. It's done at the same time, and it's a joint publication, printed and in digitally. And it's actually a tool nowadays in many countries now to assess conservation priorities. It's more tricky though for those countries don't having that, and here the global red list will mean a lot. Global red lists are also sort of at the continental level used to set conservation priorities for example, at the EU level. So it's a very good complement to national red list, and it fulfills an important task for countries don't having sort of the red list, that then they can refer to the global red list as an accepted and well-renowned document. But basically, as I said, it is this, that it's about population decline and small populations, population status and population trend, as James also mentioned. And importantly, it can be common species that is declining, as well as less common species that are declining, or very, very rare species. And in the beginning, it can look a bit tricky. How do we apply this to fungi? But I'll come back a little bit to that to give a little bit of introduction. It's fully feasible. In the beginning, it can be a little bit tricky to fully, appreciate or understand or accept that being a scientist. But it's feasible. And importantly, I think it's the same criteria at a national level and a global assessment. There is a minor adjustment, but in principle, it's the same criteria. And we did guidelines a decade ago. So there are the guideline published with recommendation how to what kind of approximations there should be for the different terms needed to be identified in the assessment process. And what I think is, I want to emphasize is that the red list is just a start. It's not, for me, that is not the important part to do the assessment. The important part is actually to get the actions done, and to get conservation action done, to have fungi considered in conservation. We need documented, well assessed species. So the actions, as I said, is more important, and documentation there is very essential. In particular, the concrete guidelines is not sufficient with just assessing species as threatened. I mean, the decision makers then don't know how to do. So it's important to have this how to do. So red listing roughly is 2 steps: compile the information and do the assessment. And this is a little bit like being a detective. There's a lot of data needed to make this let's see, did I? Here it is, yeah. It is a detective work, really. I mean, there is sort of where we combine all kind of information we have, sort of scientific papers, experiences from field observation that is not published, data from or information from the grey literature, from biological organization clubs and so forth. And of course, sort of all amateurs' observation and knowledge of the species, both distribution, what habitat they occur in, and in their ecology. And then we also very much use environmental statistics in what habitat they occur and the trend and status of that habitat. And then it's a matter of doing a very critical evaluation of what is the status, what has the trend been, and what is the trend likely to be into the future. And the higher impact the red list is getting, the more important it is that this is done well documented as possible. So when the politicians decides to take actions, and it starts to mean that management needs to be changed and cost money, then it's important that the data here is and the conclusions are as reliable as possible. Of course, the information should be transparent, and the assessment can be reassessed. Of course, it's not fixed. This is sort of the best knowledge, the best assessment we can do given the knowledge we have. So the data needed is sort of the distribution of the species. It is the population, the population size. In some way, the population trend and the threats. And then, of course, also, as I said, conservation action. And documentation is essential. You can go into the IUCN Global Red List and look at the documentation there. It's good sometimes, it's not always sufficient in my opinion. But I think it's to give a very brief description of the state of the knowledge. So it's sort of the distribution of species. Here is an example from Sweden, but it's the same at the global level. So a little bit of describing text and a map. And here is actually using the information we have, and also, which I think is important, to infer what is, given the best knowledge we have and the best sort of guesstimates we can make, what is the distribution of these species. And the population size is important because that is what the red list is based on. And this can be done in 2 ways. I mean, either it can be done based on sort of the distribution data, or it can be based on knowing in what habitat it's occurring, what quality that habitat needs to be, and how much of that habitat that it's present, and what the trend is of that habitat. And most assessment of fungi are based on the potential occurrence of the species in different habitats or substrates. It can also be for very specific species, number of known sites, and then considering also unknown sites, because there are always unknown sites. I mean, James discussed a little bit this precautionary principle, that if a very precautionary thinking would be to have just consider known sites. But with more and more people out and looking, more and more sites are being available. So that then the assessment will be changed over time. So it's much better to, and much more trust, it gives more trust if there could be some kind of estimation, which can be documented in the documentation, of how well known is this species, how conspicuous is this species, how much search for it has been conducted, has it been much overlooked or not. I mean, here is an example how this could be handled in a way. If we have the brittle fungi there to the right, which is well known, and it's easy to detect when it's sporulating, whereas sort of a corticoid fungi to the left there, that few not species, but people are able to identify, and most likely there is a microscoping needed. So it's a low degree of detectability. And then sort of the unknown number of sites and distribution is much, much higher. And then to consider sort of the amount of habitat. Certain species have a very wide range of habitats, has a broad ecology, or generalist, where there are other species have a much more narrow ecology. And the better we know the ecology of the species, in what type of conditions they occur, the more precise we can estimate to what extent they have a broader or more narrow range of habitat. And using that, we can give a very rough estimate of sort of the number of sites or area that the species may potentially occur at, and using that. And as long as we have a transparent way to describing how we have estimated the population size and its eventually trend, then it's fine. Then it can be revised. That's as we do in science. Sometimes we actually elaborate on this, the population individuals, and it's more, of course, more tricky. I mean, fungi are clonal, and they become fragmented into smaller units, still the same genotype, but of physical independent units. And we recommend in our guidelines that they use sort of the functional individuals. And here is an example of a very, very rare wood-inhabiting fungi that was confined to very special old growth pine that has been subjected to fire and been looked for very, very much. And that type of habitat is very rare. It's not being produced any longer. And we have the number of known sites in Sweden are about 100. It's been searched for intensively since during the last 30 years. And where it does occur, so if you see here, we don't estimate it to exist in more than a maximum of 300 sites. It may be 200 sites, but a maximum of 300 sites. And where it is found, it's typical 1 to 5 logs per site. And the site here may be 5 to 25 or 50 hectares. It's a very rare fungus. And then we treat each log as one individual. It may be fragmented in the log though. And but our estimation is that we have about 1,000 functional individuals in Sweden. But this is sort of exciting, I think, to think of this. It makes me really curious about how the different fungus species, how they look like, how they live, for example. I mean, they do have small to very large mycelial extension. They can be small and large, the individuals, and some are small-lived, like the needle fungi on the left here. They are small. They may live many years though in the needle as endophytes before the needle are shed, and the fungi produce sporocarp. They're very low competitive ability, so they are outcompeted as soon as the needles fall down to the floor, forest floor almost. whereas other sort of the ectomycorrhizal fungus like Cortinarius, the web caps, they last on the same spot for a very long time, for many decades, and they, as you may see here from the sporocarps here, a little bit like a fairy ring almost, that they can stay. They are larger and they can stay in the same place for a longer time. And there are other. Armillaria is, as you know, well known to extend for several hundred meters and can be very, very quite substantially old, many centuries. So having when you have better knowledge of the species, this is a way actually to handle, to work with individuals in the red listing. Here is a fungal individual, not individual, but an individual, or a genotype, a genet, as it's termed in botany. And this often chanterelles, for example, they have an extension of a couple of square meters, up to 10 square meters, the same genotype. But it's rare that it is distributed that way. I mean, fungi are typically fragmented, and that holds for aquatic fungi, of course, as it does for terrestrial fungi. And they become like this, and that is something to consider in the assessment. And I mean, this is, I think it's very exciting to get a better picture of this dynamic irrespective if it is in water or in soil, how the different species and their mycelia vary in size and presence in time and space. But red listing by itself is by individual species. Then to finalize here, to end up the talk, the trend is important for some of the criteria to assess the status, the trend of the species, and it often relies on the habitat or the substrate of appropriate quality, and then the reduction, of course, it needs some kind of time to be assessed from point 1 to point 2, and both. Here is something to consider, I think, is that certain species, they are confined to their substrate, which is limited in space and time, whereas other species, like soil-dwelling, and I would assume aquatic fungi as well, they are in a way unrestricted in size and thereby are potentially in age as well by their habitat. And we have given proxies in our guidelines how to handle this based on the limited amount of information that exists about generation length of fungi, which is tricky, but that those living on short-lived ephemeral substrate, we recommend to have 10 years assessment for those, and for wood inhabiting fungi, depending on how resistant the wood is, up to 20 to 50 years, and ectomycorrhizal fungi 50 years. But you can read more about that in detail in the guidelines. Then it's important to document the threats in a way, so that the one reading the assessment understand what is threatening, what are the causes for this species to be restricted or why it is declining. And then, as I said, even more important is this, to document how to counteract the decline, how to promote the species. I would say that this is the most important part of the text. So to summarize, I see fungal red listing as very important and as an very important contribution to the overall conservation work. And probably, let's see. Now, I should show this picture. There are several things affecting fungi as are affecting all other groups of organisms. And if we're looking into national red list, about 15 to 20% of the species are threatened, near threatened or threatened. And I would guess that in the end, we will end up with that at the global level as well. So I stop there and give time for questions if you have.
Thank you, Anders. This was, wow, so much information. I would have questions for you until next year, but I will put priority on questions from our participants today. And we have already some questions. So I will start with a question from Pratyasha Nath. I hope I said the name well. Which says, so happy to see the update in assessment numbers from about 600 last year to about 800 now. Among fungal red lists of different nations, I noticed that New Zealand, for example, has an extensive amount of data, though the assessment parameters seemed different from the IUCN criteria. I am curious if there could be a way for data to be usable across assessment criteria. Is it a form of quality control to not consider information from other red lists?
When we do, we consider all information that is available. So, the New Zealand data, when appropriate for species not being confined to New Zealand, that information from that red list is considered, of course. But they have several species that are endemic to New Zealand as well. And we have a couple of, we also actually have a couple of endemic species from New Zealand assessed on the global red list as well. But all information should be considered. And I think, just an extra thing there, I think as from a conservation point, I think as long, it doesn't matter if there are slight variations in how species are assessed on the natural level, as long as we get things assessed and there are preferably official documents that can be considered in the conservation process, then it makes it easier, of course, if it's conducted in the same way. But I see the goal is more important than the procedure, in my perspective. You may correct or add things there, James. Feel free.
Not really. It's you've pretty much got that there. I'd say for the red list, for getting on the red list, the procedure's important, but obviously we're doing the red list with a goal in mind, which is conservation. Well, specifically from my role, it's to estimate the extinction risk of a species, but why are we doing that? Because we want to conserve things and highlight what needs to be conserved.
Thank you. We have another question from an anonymous attendee asking, saying, aside from an increase in specialists and experts, which have frankly decreased in recent years, what further actions can be done to apply red listing guidelines to obscure fungi that do not have multi-celled mycelia life stages?
That do not have multicell. As it is now, the criteria applies to multicellular organisms, and that's how it is. And I don't know whether, do you want to comment on that? Because that's how it is.
Is there an opportunity to do something different, to develop some guidelines to be able to apply a similar methodology to these organisms?
So within IUCN, there is, or not necessarily within IUCN, but I have been on a call in recent weeks where there seems to be a bigger push as well, not just from the fungal kingdoms, but from wider views as well. In particular, one of the big pushes at the moment is for soil health, especially with regards to agriculture. And there's a lot of unicellular species that are very important for soil health, not just fungi, across other different phyla as well. We don't have a system in place. I think this slightly answers one of the other questions we'd had earlier that we didn't get round to. But the system as it is built towards multicellular biology, just because when we're talking about things going far along a thread here, like when you get to bacteria, what is an individual when it can actively change its genetic code during its inverted commas, lifetime, because it's splitting. It's so difficult to understand at the unicellular. We had to have a cutoff point for what we could actually include in red listing, just from a biological point of view. But don't expect it very soon. There might be a chance that we might have to start considering how we get an idea of conservation status with reference to extinction for things at unicellular life. How we do that is very difficult because we have to make sure it's consistent across an enormous amount of biology. But when that does actively become something that's being worked on, rather than just being some things that have been mentioned in multiple Zoom calls within a short period of time that we're all realizing we need to move on to look at, it's going to have to engage with as much of the community, particularly through the specialist groups. So being a member of the Aquatic Fungal Specialist Group will give you an expert chance to jump in to have your say on how those kind of guidelines could be built going forwards. The creation of such kind of guidelines take a huge amount of time. So the red list guidelines get updated each year. We're now on the 16th version of the latest version, but they're fiddling around with tiny little bits. It takes decades to update a new set, and it took so long after the creation of the Key Biodiversity Areas Initiative, the KBA initiative. It took years before they could come up with an agreed set of guidelines for how to define KBAs. So I would expect it would take years before we could get to a final decision on that. But the bigger pool and the earlier that people from the fungal kingdom can be involved in the decisions over what we put in those guidelines, the better those guidelines are going to be, rather than people not being engaged early. And then it just becomes a thing that's just bacteria, and archaea specialists write them, and then we try to apply fungi to that as well. And then no, if we're there at the beginning, everything gets included rather than just a narrower viewpoint.
And then another point there, which I think is important, is to reflect a little bit on how do we use our time best? And I think, to be pragmatic, what system is in use and can be used today, and work with that. And then it's another thing if we want to develop a system or get a system in place that is not operating today. And I mean, we have many fungi, macrofungi as well, all those that don't show conspicuous porocarps we know so little about. And I don't worry so much about those unknown species, because there is such a lack of documentation and communication of those species that we in fact already know sufficiently much about. So I would suggest to focus on the species where we know something about, and to get those assessed so those can be considered in conservation, rather than struggling with those that we don't have an assessment system in place yet. Even though that would be good. I mean, we want to cover the whole biodiversity and consider that in the future. I mean, you should also be aware of that. I think is important that the IUCN red list system, it doesn't cover everything. I mean, it doesn't take into account species that were very abundant in the past, and is just a fraction of what it was 100 years ago. They are not considered if they are short-lived or have short generation times or shorter generation times. We have many cases like that because the landscape in Europe or wherever in the world has changed so much in the last 50 to 100 years. And this is particularly problematic for many short-lived organisms, like in short-lived substrates, and many of the insects, they are not covered by major changes over several decades. So there may be also a need just to be aware of that. I think is good.
I can probably jump in and just add 2 tiny points to that as well. On what Anders has just said last, that is why we created the green status of species. So a brand new thing. There's fewer than 100 assessments, maybe fewer than 50, maybe even fewer than 20. I'm not sure. I need to check the website, but there's a very small number of those that have been published so far. And that is looking at seeing how depleted something is. So any species that's on the red list can be assessed for that, but you could find that things called least concern, because they're at a low extinction risk, are actually considered moderately or largely depleted on the green status assessment. So you then highlight, actually, this thing is not fulfilling its role in ecology, even though it's at low risk of going extinct. And I think the original question as well was about not multicelled mycelial species. So there are other things that aren't mycelial that are fungi that have been assessed. So we do have rusts and smuts on the global red list. And we're also some bracket fungi or crust fungi that have been assessed, so they can follow the similar methodology that Anders explained. And especially for the things that are host-specific parasites, you then have a very easy way to compare if that host has already been assessed on the global red list, especially if it's a range-restricted host as well, or there's a small number of the hosts. So there's other things and avenues to help do, especially if your species isn't mycelial.
Thank you. We have so many questions here. Let's move to the next one. Try to see if we can answer as many as possible. So we have a question from Ursula Ogenfus. And the question, and it says, thank you for the 2 great talks. How would you define the generation time in asexual fungi, or in those with more complicated replication cycles between asexual and sexual reproduction? As an example, Zymoseptoria kitritrici, or several spores types in rust species. Sorry, I got the name. James or Anders, who wants to take?
It takes
difficult, so I'll let Anders take it.
It's easier with those that have a sexual phase, but in fact, a sexual reproduction counts as well, as long as things can reproduce. So I think it's a matter of I mean, the definition of a generation time is sort of the average age of the parents of the newborn genotypes, or reproductive units, just to say. So it's a matter of finding an pragmatic handling of that I think make biological sense. I would be happy to elaborate on that in length, but the time is limited here. We elaborate a little bit on it in our guidelines, and I'm happy to elaborate on that through email or by Zoom at another time, but it will take some time.
Yeah, just the only thing that I would add is it can be very complex to calculate generation length. What Anders and Greg did in their 2011 paper was really, really helpful especially for the terrestrial macrofungi, and figuring out a nice set of replicable generation length that we used across multiple species. But that isn't available to most of the groups of species that are assessed on the red list. So within the red list guidelines themselves, which you can download from the red list website, there is a long section on generation length and all the multiple ways that they can be calculated. So if you have data that would fit into one of those methods, then you can use any of the methods that are available. And within the assessment, there are boxes that allow you to fill in how you calculated generation length to justify your decisions, etc.
And this is just an extra thing, but this makes it very fascinating, I think, to reflect upon how the genes are moving in time and space for different species. That's exciting.
Thank you. We have another question from Andres Bruder, kind of fits here, and he's asking, in assessment efforts, should we invest in relatively costly molecular techniques, such as population genetics, to better resolve between functional individuals and genetically different individuals of the same species?
I think, in my perspective, we get very good sort of general information from molecular data and genetic analysis. But often, put it this way, if we can get better proxies, better understand whether we have single genotypes, or, but then to do it properly, we would need, in a way, to approach a better understanding of how the number of units, the ramets and their dynamics in a site, whether it's aquatic or terrestrial. I mean, there is a difference if we just are thinking of monitoring occurrences. Then there is a difference in aquatic environment and terrestrial environment. I mean, aquatic, you have a better mixture and can better detect rare things. In terrestrial habitat, it's I would say we're just detecting, even with numerous samples, we are detecting all common species. The rare one in the tail will never be detected. Still, it gives very important can give complementary information about common species, their presence and so forth. But it's I think the combination of traditional methods and the molecular methods is the way ahead. But of course, it gives better information about the biology, and that is critical to better assess, better understand species and how to counteract sort of decline and so forth.
Thank you, Anders. We have another question from an anonymous participant that says, have you experienced any issues with ecological data that you know exist and is relevant, but is not accessible to the parties doing the red listing? And if so, how did you deal with this?
I didn't, sorry, I'm slow thinking. I didn't really get that question. What was asked for?
Well, I cannot call the person itself.
No.
I don't have the name here, but basically the person asks what can happen if there is some data that might exist, but is not accessible for the people doing red listing. I don't know, it can be, for instance, maybe people don't have access to the papers which are published, or maybe some gray literature that is not available to everyone to have access to it. I'm just figuring out that this might be the question behind.
Yeah. I mean, I think it's a matter of being pragmatic again. It's like deciding with the family where to go on holiday. We don't have everything on hand. Where are the best places to go with the family and the kids and so forth? It's the same here. We're using the information that is available, and we do the best assessment we can. I mean, that deals with, for example, occurrences. If we do that, it's tricky on the national level to know the distribution and the occurrence, its frequency, how frequent it is in different habitats, and the trends. And then scaling that up to the European level or to the global level is more tricky. It's not easy to get access of data, what is going on in Russia, for example, or in China. And different countries have different knowledge of distribution and trends. So I think it's I wouldn't worry. I'm not worrying about that, but the more information, the better information we can get, the better it is. But that is as having sort of a scientific approach that we are documenting on what basis we do the assessment. Then it can be reassessed. So I don't. For me, it's not the problem.
Thank you. We have one more question from Hadi Karut. Apologies if I'm spelling your name wrongly. And they're asking, are there any considerations for fungi conservation in Arab countries?
Yeah, I mean, there is a Arabic Society for Fungal Conservation. There is such an organization, as you may be aware of, terrestrial fungi. And they have taken part in some assessment. It hasn't been so much activities yet, but I know that, for example, the desert truffles, the Terfesia, has been there've been attempts to finalize them, but as it's in principle it's based on voluntary actions to 100%. It depending on where there are individuals and groups that have the interest and time to contribute. So it hasn't been much yet, but there is a need there, as in any part of the world.
And of course, in areas where there hasn't been as much effort, that leaves a bigger hole for someone to make a big impact by going out and being the one that pushes things forward and gets more of those assessed and then can push forward the conservation through there.
Good point.
So we have one last question on our Q&A, and it's from Ricardo Dreschler. Dear Anders, thank you for your talk. I appreciate so much. As you said, there are many tricks to apply IUCN criteria for fungal species, and your work with Greg is great and help us a lot, mainly with those macrofungal species. Do you have plans to produce some guidelines for other groups of fungi, mainly those micro, aquatic, etc.? We kind of already touched this question, but I wanted to still raise it up because being part of this new IUCN Aquatic Fungi Specialist Group, my answer to this question is yes, we want to make sure that we do all the efforts possible to apply red listing also to aquatic fungi. And if new guidelines are needed, we are willing to work on that. But I will also want to hear from Anders and James about how maybe we can collaborate together to produce this.
And we have had the intention for a while, Greg and I, to update and be more inclusive with the guidelines, and that could be by collaborating with you, for example, to have to update it and to make it to include other groups of fungi as well. So the plans are there, but the time hasn't been there yet. But you may help us to do that and contribute.
We will be glad.
Also, from a clonal species point of view, so this isn't just fungi, but all clonal species, so going to bryophytes, corals, what have you. There's a need to standardize that across all of the different groups, so that the red listing is once again truly a global thing in terms of it can be applied to all species. So there is a move towards trying to standardize that, and hopefully through the red list technical working group that I represent fungi on, there's going to be, we've been promised it for a while, that there's going to try and be a more all-encompassing discussion group with the specialist groups that represent all of those clonal species, so that we can all come together and come up with a standardized approach towards red listing within those groups, so that it properly reflects everything, and so people can use the same categories criteria across all clonal organisms.
Thank you so much. I think we're reaching the end of our webinar. Thank you, Anders and James, for those two amazing presentations. Thank you for my co-hosts, and thank you for everyone who joined us today. As we mentioned in the chat, we have recorded the webinar and we will share it in the FUNACTION YouTube channel. We will also send an email to everyone who registered as soon as the video is available online. And please do reach out to us if you have any further questions, or if you would like to be more engaged on red listing or just the SSC specialist groups in general. And keep an eye for further webinars. We will be organizing one very soon on key BAs, key biodiversity areas, and we will advertise it in the FUNACTION and Global Center for Species Survival social media. So do keep an eye, and thank you so much. I don't know if anyone has some final thoughts. No?
Well, if not, thank you so much for attending, and see you in the next webinar. Thank you.
Thank you. Bye.